Yeah, so the idea here is that, as I just said, conventional T cells mostly fail to penetrate, infiltrate solid tumors, or even to thrive. They're the ones that make it there. But there's a subset of T cells that do infiltrate and get enriched in solid tumors. That's a regulatory T cell. So this is a subset of CD4 T cells, roughly 5%, and their function in the body is to maintain autoimmune attacks from being away. So if you have Tregs working, there's an autoimmune disease and unwanted rejection. So usually Tregs then are bad news for tumors, right? So solid tumors that kind of learn immunology and they enrich themselves with Tregs to keep any immune surveillance at bay. What I found, however, is that if you modify a human regulatory T cell with a chimeric antigen receptor, a CAR, directly targeting a tumor cell, and I do an in vitro killing assay, these CAR-Tregs actually kill the tumor cells. And if you put them in NSG mouse, so you put human tumor cells and human CAR-Tregs in immunodeficient mouse, the CAR-Tregs, they control the growth of tumor in NSG mice. And so what we're looking now more molecularly, what we find is that activation of a human Treg via high affinity CAR induces inflammatory cytokine production, the cytotoxic molecule production, all while maintaining the Treg chemokine receptor expression pattern. Because, for example, CCR8, Tregs express this cytokine, this chemokine receptor very highly, T effectors don't. And for pancreatic cancer, for example, this chemokine receptor has been shown to be important in infiltration. So we're making pro-inflammatory CAR-Tregs. So they acquire some functionalities of T effector cells while remaining Tregs. So they have FOXP3, they don't produce IL-2, and they maintain their chemokine receptor expression pattern.